Over an aerial view of the Life Sciences building, text reads, University of Dundee, School of Life Sciences. Understanding how the immune cells work. In a lab, a scientist places a 6-well plate under a microscope. One of the wells contains pink-tinged liquid media. The scientist looks through the eyepiece.
Our laboratory studied T lymphocytes, also called T cells. They are a type of white blood cell and form part of our immune system. They help protect the body from infection by killing cancer or infected cells. In our experiments, we use T cells that are found in the lymph nodes, spleen, and blood. We keep them in plastic plates and flasks filled with liquid media that contains all the nutrients they need.
A labeled, microscopic image shows naive T cells.
These T cells are called naive. They are small and not very active. However, during an infection, they are activated and convert into effector T cells.
The image changes to show effector T cells. The cells on screen change from small, round dots, and become larger and more irregular in shape. The scientist removes the lid of the 6-well plate and pipettes an activator into the well. She places the plate into an incubator.
For some of our experiments, we convert these naive T cells into effector T cells. This lets us study how they act during an infection. To do so, we activate naive T cells using an activator that we add to the plate. We place them in an incubator set at 37 degrees Celsius and 5% carbon dioxide and leave them for 48 hours.
Microscopic footage shows naive T cells changing into effector T cells. The scientist removes the 6-well plate from the incubator. The liquid media in the well has turned yellow.
During this time, the T cells grow in size and rapidly increase in number. After this time, the liquid media has turned yellow, meaning that the cells have used all the nutrients present in it.
The scientist studies the cells under a microscope. She then removes the yellow liquid media from the well. She places a tube containing the T cells suspended in liquid into a centrifuge.
We collect the cells by spinning them in a centrifuge and put them in a flask with fresh media to provide more nutrients for their rapid growth and increase in number.
She extracts a red liquid media from a bottle, and then draws the cells from the centrifuge tube and adds them to a flask. She draws IL-2 from a smaller tube, and pipettes it into the flask. The flask is labeled, effectors.
We add a cytokine called interleukin-2, IL-2. This helps T cells to convert into effector T cells and increase in number further.
The scientist gently mixes the contents of the flask, then places it into an incubator.
We leave them in the incubator overnight.
The scientist removes the flask from the incubator and studies it under the microscope.
In the morning, the cells have grown and divided, making millions of these killing cells. Now we count them.
The scientist extracts a sample from the flask and pipettes it into another tube. She injects a clear liquid into a 15 milliliter tube before adding it to the first tube.
We prepare a sample for counting by adding 100 microliters of our liquid media to a tube. We go to the bench and add 900 microliters of PBS containing DAPI to the T cells. DAPI is a fluorescent DNA stain that only stains dead cells. We count the T cells on a machine called a flow cytometer. It can count up to 10,000 cells per second.
The scientist retrieves a packet of flasks and labels another flask as effectors D5.
We grow or expand these cells for a further 5 days, keeping them at a density of 0.3 to 0.5 million cells per milliliter.
The scientist pipettes the pink-tinged liquid media into the flask labeled effectors D5. She extracts IL-2 from a small tube, then adds it to the flask. She draws liquid media containing effector T cells from the original flask, then injects it into the second flask. She gently rocks the flask.
As they keep increasing in number, we need to do daily counts and replace the liquid media containing IL-2. The cells are stored in an incubator during this time. Now we know how many T cells we have, we can use them in all sorts of exciting experiments.
She draws a small amount of liquid media from a flask labeled effectors D6, then pipettes it into 2 wells of a 96 well plate. She places a lid over the well plate, and gently moves it around with her hands.
We can study how they respond to different infections, and how nutrients and their environment affect their ability to fight infection.
The scientist adds a tube to a flow cytometer which she operates from a computer. A probe goes into the tube. Data appears on the computer screen as scatterplots. In an office, she writes notes as she works at another computer.
One thing we can look at using the flow cytometer are differences in the features of the T cell. We can compare the size of naive and effector T cells. Effector cells are much bigger in size compared to naive cells. They are also more granular. This is because they have specialized granules containing granzymes used to kill the target cells.
2 scatter plots appear on the computer. Below each is another graph.
The data from the flow cytometer is shown as scatter plots. Forward scatter, FSC, is used to measure cell size, whereas side scatter, SSC, indicates the granularity.
Back in the lab, the scientist pours the sample into a liquid waste bottle and seals it, before gently spinning the contents.
Once we have finished our experiments, we throw away our cells into liquid waste. Wellcome, a biocide used to destroy biological material, is added.
A black letter W appears over text to form the Wellcome logo. Text below reads, supported by the Wellcome Institutional Strategic Support Fund awarded to University of Dundee. A blue and white crest appears beside blue text to form the University of Dundee logo.